This protein was designed in 6 hours. It binds to the malaria parasite 10,000 times better than anything nature made in 4 billion years. This battery electrolyte was drafted in 3 days. It used to take 18 months of trial and error. This catalyst was tested in a weekend. It turned CO2 into fuel at room temperature. We didn't find these molecules, we designed them. On purpose. For centuries, we searched for useful materials the way you'd search a library with no catalog. Pick a molecule, test it. Wait months. Try another. Fail. Repeat. Penicillin? Accident. Teflon? Accident. Even the transistor took decades of wandering. Then came the turn. Instead of asking, what does this molecule do? We learned to ask, what do we need? And we taught computers to answer. Here's the loop that changes everything. Specify. State exactly what you want. Conduct ions like a liquid. Stay solid, don't catch fire. Generate. Let AI draft 1,000 candidates in hours. Models trained on millions of molecules propose structures that could work. Screen. Filter ruthlessly. Physics checks. Cost estimates. Safety rules. Most fail here, that's the point. Refine. The lab tests the survivors. Each result teaches the model. The loop tightens. The next round is smarter. Pixels become pipettes. Pipettes become proof. 3 things converged. First, we have databases. Millions of known molecules and their properties. The training data exists. Second, we have models. AlphaFold cracked protein folding. Graph neural networks learn to read crystal structures like sheet music. Third, we have infrastructure. Self-driving labs that run experiments while we sleep. Robots with gentle wrists. Sensors that measure in real time. The method isn't new, but the speed is. What took a career now takes a coffee break. In the next hour, you'll see this loop work across 4 domains. Batteries that can't catch fire. Proteins designed to cure disease. Catalysts that clean the air. Materials that last decades. You'll meet the labs where robots run the experiments. The guardrails that keep this power safe. And the trade-offs we're making right now. Because speed without wisdom is just faster failure. Discovery used to wander. Now, design chooses a horizon and walks. Your phone battery is a controlled bomb. Liquid electrolytes conduct beautifully and burn spectacularly. Lithium metal anodes pack twice the energy, but grow needle-like dendrites that pierce the separator and short-circuit the cell. We've known the solution for decades. Solid electrolytes. A highway for ions. A wall for electrons. No liquid to burn. The problem? Most solids conduct ions like molasses flows uphill. So we wrote a spec. Ionic conductivity, 10 to the -3 to 10 to the -2 Siemens per centimeter at room temperature. Stable against lithium metal. No reactions, no dendrites. Survives humidity, doesn't dissolve on a Tuesday. Made from abundant elements, no unobtainium. Thin film compatible. We need to print this. This is the target. Now we ask the computer, show me candidates. The model proposes frameworks. Garnets, cubic crystals with wide channels where lithium ions sprint like trains through tunnels. Nasicons, 3-dimensional networks with multiple pathways. If one route clogs, ions find another. Sulfides, the fastest conductors, but they don't like air. Polymer ceramic hybrids, Flexibility meets durability. Each suggestion comes with predictions. Formation energy, ionic mobility, stability windows. The model doesn't guess. It calculates from quantum mechanics, condensed into fast approximations. 1,000 candidates in an afternoon. Now the culling. Physics filters catch the impossible. Valence errors, unstable phases, structures that collapse under their own symmetry. Cost filters catch the impractical. Rare elements, synthesis that needs a star's core temperature. Safety filters catch the dangerous. Toxic byproducts, reactivity with water. Of 1,000 candidates, maybe 50 survive. The shortlist goes to the lab. Powders are mixed, pressed into pellets, fired in furnaces with atmospheres tuned to the 10th of a degree. Impedance spectroscopy measures conductivity. Cycling tests measure endurance. Electron microscopy looks for dendrites. Most still fail, but each failure teaches the model. Too brittle at this grain size. Conductivity drops in humidity. Dendrites nucleate at grain boundaries. The model updates, proposes modifications. Dope with magnesium. Add a buffer layer. Change the sintering profile. The loop tightens. By round 3, you have something that works. A solid-state electrolyte that conducts at room temperature. Stable for 1,000 cycles. Safe enough to bend, puncture, even cut. And it won't catch fire. From spec to prototype, 6 months. The old way, 5 to 10 years. A material is real when it survives Monday. And this one does. A protein is a sentence that folds into meaning. Start with a string of amino acids, a one-dimensional code. It drifts in water, testing thousands of shapes per second. Then suddenly, click. One configuration. Stable. Functional. Shape is how a protein speaks. Binding is how it keeps its promise. A groove that fits a partner molecule. Charges that attract at exactly the right distance. Loops that flex to hold, then release. This is molecular recognition, the most precise language in nature. For 50 years, we could read the DNA code, but not predict the shape. Thousands of person-years spent crystallizing proteins, shooting X-rays, solving structures one at a time. Then, in 2020, AlphaFold. Given only the amino acid sequence, it predicts the 3D structure. Not just close, nearly perfect. Atom-level accuracy in minutes. But AlphaFold 1 only predicted single proteins. Biology doesn't work that way. Life is a crowded stage. Proteins work in teams. They bind DNA, grab metals, partner with other proteins in precise orientations. AlphaFold 3 predicts the whole ensemble. Protein-protein complexes, protein-DNA interactions, small molecules docking into active sites. And it tells you how confident it is. Blue regions? Certain. Yellow? Maybe. Red? Don't trust this part. For the first time, we can design the interaction, not just the structure. Let's design a biosensor binder. Target: one specific protein in a messy sample. Ignore everything else. Specify: nanomolar binding affinity, tight enough to hold on. 100 times selectivity over similar proteins. Recognize the right face in a crowd. Stable in salt water, body temperature, realistic conditions. Generate. We don't start from scratch, we start from scaffolds. Small, stable protein frameworks known to be well-behaved. The model proposes sequences. Try these amino acids in the binding pocket. This loop should flex here. Add a negative charge to repel off targets. Thousands of candidates. Screen. Predict binding to the target, strong signal. Predict binding to off targets, weak or none. Predict stability, survives heat, doesn't aggregate. Check for red flags, unusual motifs, potential toxicity, manufacturing headaches. Down to 50 sequences. Refine. Synthesize the top 10 as DNA. Bacteria express them overnight. Test binding with surface plasmon resonance. Most are mediocre. A few are promising. The model learns from the winners. Aromatic residues in this position improve specificity. Polar residues here stabilize the loop. Round 2, even better. Round 3, a binder that works. From concept to working protein, days, not months. Remember the opening? A protein designed to bind the malaria parasite. Real example, real lab. They specified, grip this surface protein on Plasmodium falciparum. Ignore human proteins. The model proposed binders. After 3 rounds of testing and refinement, they found one with picomolar affinity, 10,000 times stronger than anything nature evolved. Why? Because evolution optimizes for survival, not for our therapeutic goals. We optimized directly for binding. No compromises, no evolutionary baggage. The binder is now being tested as a biosensor for rapid malaria diagnosis. Drop of blood, 30 seconds, result. But here's the tension. If we can design proteins that bind anything, we can also design toxins. Proteins that disrupt essential pathways. Binders that shouldn't exist. So we built guardrails. Sequence screening. Every design checked against databases of known toxins, venom proteins, restricted sequences. Off-target prediction. Does this bind anything it shouldn't? Tiered access. Benign predictions are open. High-affinity binders require approval, audit trails, human review. Red teams. People paid to try to design dangerous things. When they succeed, we patch the holes. The lab is the last word. Structure predictions are hypotheses. They mean nothing until cells, tissue, and animal models agree. Speed is a tool. Responsibility is the hand that holds it. Crystals are cities. Their atomic arrangement is the street grid. Change the layout, change what flows through. Garnets, wide avenues where lithium ions sprint. Perovskites, choirs of atoms that absorb light and sing electricity. Moths, porous palaces with rooms sized for specific guests. We used to stumble on good layouts by accident. Now we draft the city plan first, then build it. Graph neural networks read lattice structures like architects read blueprints. Nodes are atoms, edges are bonds. The network predicts, stable or unstable, conductor or insulator, brittle or tough. Perovskite solar cells hit 26% efficiency, rivaling silicon. But they have a problem. They hate weather. Humidity dissolves them. Heat cracks them. Light itself degrades them over time. We need 1,000-hour stability, real roofs, real rain. The loop. Specify. Maintain 90% efficiency after 1,000 hours of light, heat, and humidity. Use non-toxic elements. Generate. The model proposes cation mixtures. Cesium for stability. Formamidinium for efficiency. rubidium to calm grain boundaries. It suggests 2D/3D hybrid structures, layered like a laminated windshield. The 2D layers block water. Screen. Predict band gaps. Defect formation energies. Decomposition pathways. Refine. The lab makes films, stress tests them, adjusts the recipe. Add a passivation layer. Tune grain sizes. Encapsulate better. The result? Cells that pass damp heat testing. Efficiency holding steady after months. Still not perfect, but getting there. And when they cross the threshold, when they last a decade on a roof, that's when solar becomes infrastructure, not experiment. Metal-organic frameworks are skeletons of metal ions connected by organic linkers. The result? Crystals filled with empty space. Porous, organized, selective. You can design pores that prefer one gas over another. CO2 over nitrogen for carbon capture. Methane over water for natural gas purification. The challenge? Most MOFs collapse in humidity or cost a fortune to make. The loop. Specify. Capture CO2 selectively even when wet. Use cheap metals. Regenerate at low temperature. Generate. Models propose thousands of framework topologies. Different metals, different linkers, different pore sizes. Screen. Predict binding energies for CO2 versus N2, stability in water, synthesis cost. Refine. The lab synthesizes the finalists, tests uptake, cycles them, learns which linkers tolerate moisture, which frameworks stay rigid. The survivors, MOFs that work in real flue gas, not just dry lab air. From concept to working filter, months, not years. Electrocatalysts turn electricity into chemistry, water into hydrogen, CO2 into fuels, nitrogen into ammonia. The problem? Most reactions need brutal conditions or precious metals. Hydrogen evolution. Platinum works beautifully. Also costs $30,000 per kilogram. CO2 reduction. We want one product, ethanol, ethylene. Instead, we get a mess. Methane, formate, carbon monoxide, all mixed together. We need catalysts that are selective, efficient, durable, and cheap. The loop. Specify. Convert CO2 to ethylene with 80% selectivity. Run at low overpotential. Last 500 hours. Use earth-abundant metals. Generate. Models propose surface structures. Copper alloys doped with zinc. Specific crystal facets. Defect sites engineered for binding. Screen. Predict binding energies for intermediates. Calculate overpotentials. Check for poisoning pathways. Refine. Electrochemistry labs test surfaces. Measure Faradaic efficiency. Watch for degradation. The model learns which active sites work, which ones poison, which ones last. The result? Copper-based catalysts that approach industrial viability. Not perfect, but 10 times better than 5 years ago. And that's the pattern. Not miracles. Progress. Here's a subtle turn. Perfection isn't always the goal. Sometimes you want controlled imperfection. A vacancy, one missing atom, can be a shortcut for ions. A dopant, one wrong atom, can change conductivity by 1,000,000 times. A color center, A tiny defect that traps light becomes a quantum sensor. Nitrogen vacancy centers in diamond detect magnetic fields at the single neuron level. Not in an MRI machine, on a benchtop, at room temperature. We don't just design the crystal, we design where it's imperfect. And the loop learns this too. A fingertip taps a screen. Across the room, a robotic arm moves. A pipette draws liquid. A reactor warms. Spectroscopy measures in real time. The data updates the model. The model chooses the next experiment. This is a self-driving lab. The human sets the goal. The system explores. Not autonomy, augmentation. The robot does the repetitive work. The scientist does the meaningful work, asking which questions matter. Here's the trick: active learning. Instead of testing randomly, the system asks, which experiment teaches me the most? It looks for the borderlands, where the model is uncertain. One measurement there redraws the map. 10 strategic experiments beat 100 random ones. Example: you're optimizing a catalyst. Hundreds of conditions to try. Temperature, pressure, dopant concentration. Random sampling? You'd need months. Active learning? The model tests a few points, learns the landscape, predicts where the peak might be, tests there, updates. 5 iterations, 20 experiments, found the optimum. The full closed loop. 1. Propose. Model suggests conditions. 2. Make. Robots synthesize or prepare samples. 3. Measure. Inline sensors collect data during the reaction. 4. Learn. Model updates, refines predictions. 5. Repeat. Next proposal is smarter. No human waiting for results. No delays between batches. The loop runs overnight. Results. Perovskite stability doubled in a weekend. 288 compositions tested. Solid-state electrolyte conductivity improved 3 times in 4 days. MOF synthesis optimized. Yield up, cost down. 150 conditions explored. From months to days. But humans stay in the loop. We decide what questions matter, what good enough means, when to stop, what's safe to try. We design the constraints, the stop rules, the meaning of the data. The lab automates the waste, repetitive pipetting, overnight reactions, 24/7 monitoring. It doesn't automate the wonder. When a result surprises, a color that shouldn't exist, a reaction pathway nobody predicted, that's when the human eye matters. The lab extends reach. It doesn't replace judgment. These aren't visions, they're running. Closed-loop battery optimization, operating at national labs. Protein design platforms, used by pharmaceutical companies. MOF screening facilities, testing carbon capture at scale. Catalyst discovery loops, finding electrolyzers for green hydrogen. First prototypes are clearing their throats. Not products yet, but proving grounds. The question isn't, will this work? It's, how fast can we make it work everywhere? A material is real when it survives Monday, but a product is real when it survives 1,000 Mondays. Discovery happens in a beaker. Deployment happens in a factory. And the gap between them is brutal. On the bench, 1 perfect run. Controlled conditions, patient hands. In the plant, 100 runs per shift. Dust, heat, variability, maintenance schedules, supply chains. Scaling is not copying, it's translation. The bench celebrates a result. The plant asks, how wide is the window? Not does it work, but how far can conditions drift before it stops working? Temperature plus or minus 5 degrees Celsius? Humidity 30 to 60%? Impurities under 100 parts per million? You map the window. Then you write the recipe so it stays inside. Example: solid-state electrolyte. The bench version. Mixed at exactly 800 degrees Celsius for exactly 12 hours in ultra-pure argon. The plant version. 790 to 810 degrees Celsius for 11 to 13 hours in 99.9% argon, because ultra-pure costs 10 times more. You test the edges, learn where it breaks, build margin. The tighter the window, the more expensive the control. The wider the window, the more robust the product. Design for the Tuesday version, not the perfect Sunday. You don't go straight from bench to billion-dollar factory. First, the pilot line. 10 times bigger than the bench. 1/10 the size of the plant. It's a truth machine. It finds the mixing step that doesn't scale, the filtration that clogs after batch 3, the coating that's uniform in a Petri dish but patchy on a roll. Each problem caught here saves millions later. You learn, you adjust, you lock in the recipe. When the pilot runs for a month without surprises, then you talk about factories. A material isn't ready if its precursors come from one mine in one country. You need second sources. You need stockpiles. You need substitutes designed before the shortage. So the loop learns this too. Prefer abundant elements. Avoid single-source materials. Design with degradation pathways that recycle cleanly. Resilience is part of the spec from day one. Faster design is good. Faster design of disposable junk is not. So we count energy input per kilogram, water use, toxic byproducts, end of life, recyclable, biodegradable, or landfill. The model learns to prefer materials that last longer, processes that waste less, chemistries that close loops. Because speed without sustainability just accelerates the problem. Good tools need good rules. Here's the framework we're building. R.A.I.L.S. R. Record. Every design has a lineage. Dataset used, model version, prompt, parameters, time-stamped. Append-only logs. If a result matters, you must be able to reproduce it. If a result fails, you must be able to learn from it. A. Audit. Independent review. Model cards. What it predicts well, where it fails, where not to trust it. Datasheets. What trained this model? What's missing? What biases exist? Red teams. People paid to try to break the system. When they succeed, we fix it. I. Interlock. Tiered access based on risk. Tier 1. Open. Benign predictions, materials databases, educational tools. Tier 2. Gated. High affinity binders. Synthesis routes for potent compounds. Scale-up protocols. Requires approval, audit trail, rate limits, human review. Tier 3. Restricted. Anything with dual use potential. Requires institutional oversight, ethics board, regulatory consultation. L. Label watermarks and confidence. Every AI-generated structure carries metadata. Origin, model, confidence score. If the model is 95% confident, you can trust it. If it's 60% confident, verify in the lab first. Humility embedded in the output. S. Share. Safe methods. Near misses. Publish negative results. Share datasets when safe. Document where the model failed, because collective learning is faster learning. These aren't hypothetical. They're being implemented now. Protein design platforms. Tiered access live. Materials databases. Provenance tracked. Self-driving labs. Audit logs mandatory. Not perfect, but present. Where does this go next? 1. Medicine that fits. Not one drug for everyone. Binders designed for your variant of a disease protein. Enzymes tuned for your metabolism. Personalized at scale. 2. Materials that adapt. Self-healing polymers. Coatings that sense damage and repair. Alloys that change properties on demand. Not static. Responsive. 3. Carbon capture that works. MOFs that pull CO2 from air cheaply enough to matter. Catalysts that turn it into fuel, or concrete, or plastics. Not offsets. Reversal. 4. Fusion and fission materials. Plasma-facing materials that survive neutron bombardment. Coolants that don't corrode under radiation. The engineering problems holding back clean energy. These aren't science fiction. They're engineering problems with defined specs. And now we have a loop to solve them. The bottleneck used to be ideas. Now, it's verification. We can generate 1 million candidate molecules overnight. We can only test 1,000 per year in the lab. So the next leap is making the lab faster. More throughput, better sensors, cheaper synthesis. The loop is fast. The lab is the constraint. But that's a good problem. It's an engineering problem. We solve those. But here's what keeps me up at night. Who decides what gets designed? Right now, mostly wealthy institutions, big companies, well-funded labs. The loop is open source. The compute is cheap. But the priorities, those are still set by whoever writes the checks. Will we design malaria binders, or just more profitable blockbusters for wealthy markets? Will we optimize for planetary health, or just for quarterly earnings? The tools are neutral. The choices aren't. We taught sand to dream molecules into existence. We built labs that learn while we sleep. We turned design into a loop that tightens every day. This power is here. The question is, what do we build? Guardrails aren't obstacles. They're how we move faster, safely. Transparency isn't weakness. It's how we build trust. Humility isn't doubt. It's how we stay honest. We did not leash atoms. We taught them a language. Now we need to choose our words carefully. From pixels to pipettes, from models to matter, we designed a battery that won't catch fire, a protein that binds malaria, a catalyst that breathes CO2, a solar cell that survives weather. Not by luck, by loop. Specify, generate, screen, refine. This is the method. It works today, And it's accelerating. You don't need to build the tools to use them. The databases are open, the models are published, the notebooks run in browsers. You can sketch a molecule this afternoon. You can compare materials tonight. You can propose something new by the weekend. The future isn't found. It's drafted, tested, and shared. Skill is a kindness you can offer the future. Discovery used to wander. Now design chooses a horizon and walks. Crystals are cities. Their symmetries are streets. And we learn to draft the skyline before laying the first stone. Proteins are sentences that fold into meaning. Shape speaks. Binding keeps its promise. And we learn to write new words in an ancient language. The lab became a loom. Data became the thread. Insight became the cloth. A material is real when it survives Monday. A miracle is real when it becomes ordinary. When batteries that don't burn are boring. When proteins designed in hours are routine. When materials optimized by AI are just materials. That's when we'll know we succeeded. We took a grain of sand and taught it to remember. We asked it to imagine. And it answered with matter that works. This is Cosmic Canvas, where science meets story. Where the universe isn't just studied, it's painted in meaning. If this moved you, leave a comment. Tell me which discovery surprised you most. Tell me what you want designed next. We didn't leash atoms. We taught them a language. And the conversation has just begun.